Hello, my name is René Ranzinger, and I'm going to introduce the GlyGen project to you. GlyGen is a database or a knowledge base that integrates glycomics and glycoproteomics data from various resources into a single database that people can access and use project. It's a large project with lots of different people involved. The major groups are at the University of Georgia with Mike Thiemeyer and the George Washington University with Raja Masunda. But we also have a lot of collaborators at the Harvard Medical School, Soka University in Japan, Griffith University in Australia, Imperial College in London, the Swiss Bioinformatics Institute, people at NIH, AMBL, EBI, working with us together on that project. So what is GlyGen? I already said it's a data integration project. There are multiple different databases out there that store glycomics and glycoproteomics information. And all of these databases have valuable information, but they are not very well connected, especially if it comes to connecting the glycomics and proteomics part with each other. What we are doing is we are accessing all these resources. We download the data and we standardize the data in standard formats. We do a quality check on the data, and then we convert this into one single database, which now contains the glycomics and glycoproteomics data in one system. And on top of that, we are creating multiple interfaces, or we have created multiple interfaces that allow to access the data. There are several of them for bioinformatics use. So there's a Sparkle endpoint, there's a web service API, and there's a data store. But we also provide interfaces for the use via the browser, where a normal person can go using the web browser and explore that data. And that's what I'm going to focus on in during my talk, to give you an impression on that web portal and how it works. So all the data is really available at the glygen.org domain. You can just go there, you can play around with that. And that's exactly what I'm going to do. I'm going to give you a live demonstration of the web portal using the browser. Okay, so that's one of the pages from the web portal. What we are looking at right now is a GlyCan page, just to give you an idea what kind of information we retrieve from these other resources and which we store in our system. So let's quickly go over that page. In the beginning of the page, we have the glycan cartoon, the glytucan accession number, the mass, the composition, the glycan type, and so on as the general information. And while you're scrolling down, we see additional information such as organism, like in which organisms has that glycan being reported. What you can see here is these little colorful badges which indicate from which database we retrieved that annotation. Like which database said that this glycan was reported in human. There are 3 of them, GlyConnect, GlyTucan, and UniCop. And if you click on these badges, they expand and they show the database entries in that corresponding database which provided that annotation. By clicking on that link here now, you can actually go to that database and see the information of that structure in that database, and also the annotation that they provided to us. And that's true for pretty much all information in our system that we retrieve from other databases. They are always tagged with that badges to allow users to judge which system the information came from, or even go to that other database to get more detailed information. In that sense, GlyGen is not just a database to view information, but it can also serve as a hub to find more information about that glycan or that protein in these other databases. But let's continue with the glycan. We have a list of motifs that we are checking against each glycan, and we report which of these motifs have been found in that glycan. We have the proteins that have been reported carrying that glycan. So for each protein, again, we see the resources that provided that annotation, the protein name, the accession, and if known, the position on the protein where the glycan has been reported. If binding of protein to the glycan is reported, it can be shown, not present for this particular glycan. We have enzymes for that glycans that may have been involved in the synthesis, the biosynthesis of that glycan, also from different species. So we have mouse here, we have human enzymes. We have related glycans which share the same composition or the same topology. We have expression information, like in which tissues or in which cell lines has that glycan been reported on. Digital sequences, cross-references to other databases. There's a large variety of databases that we are linking to, and by clicking on each of these links, you can go to these databases. So there are multiple CardBank entries, KEGG, glycosciences.de, but also KB, PubChem, more chemical databases. And then in the end of the page, we have a very long list of publications in which this glycan has been reported. Okay, so that's about the glycan. Let's go to the protein. So for the protein page, we are getting a lot of information mainly from UniProt, but also from other resources. So in that general box, which is similar to glycan, we have different names and accession numbers for the protein. Also, if known, the chromosomal location, and of course the species that the protein comes from. Then next we have the glycans that have been reported on that proteins. There are multiple different ways how that can be reported. So there can be glycans, there can be sites where a particular glycan has been reported, but we also have proteins where just the site is reported to be glycosylated, but it was not reported which glycans are on there. For some proteins, we also have predicted sites, and we also have a collaboration with a text mining initiative to extract information out of papers, and we report additional sites. Different names for proteins, protein function, the sequence of the protein. I will go into that in detail a little bit later. If there are mutation information have been reported, we show them, either if they are disease related or if they are not disease related. Mutagenesis data, Go annotations. If the protein itself binds a glycan, we can also see the glycan structure that is has been reported to be bound by that protein. We have pathway information, like in with pathway that protein is involved. If the protein itself is an enzyme, you will also be able to see the list of glycans that are synthesized or may have been synthesized by that proteins. We have a list of isoforms and homologs in the different species. If the protein is involved in any kind of disease, there is a summary of that section as well. We have expression data in different tissue for that protein, or also expression data, how the expression changes for diseases. A cross-reference section which lists all the proteomics databases that have web pages for that particular protein that we are looking at. And then in the end, also, we have a publication section with the list of papers that talk about that protein. I'm going back to the glycosylation section just to illustrate how the database is a little bit different from like UniProt. So on UniProt, you will not have the information which glycan is on a particular position. You very often just have that a site is glycosylated, but they don't know which glycan is on there. So, but we retrieve from other databases that information, like Connect, for example, or UniProtKB, that reported that and bring this together. So that's why you can find on the glycan side more information than you would actually be able to find on UniProt. What you also can do is you can click on these glycan IDs and immediately go from the protein page to the corresponding glycan page, which then shows you the summary of the glycan. And similar on the glycan side, you can click on the protein ID and then go to the protein or one of the proteins that have been reported carrying that glycan. Now, that's what we store for glycans and proteins, but how can you find the glycans and proteins that you're looking for? So we have multiple different searches that we provide under the Explore menu. There's a glycan search, where you can go and search by different glycan properties, by ID, by mass, by organism, and so on. We also have a composition search, and we have a similar search field for the proteins that you can use to access the proteins. But the most advanced feature that we have in terms of searching is something we call a super search, which represents all the information that we have in the database in a graphical way. So you can see we have motifs, there are a total of 120 motifs. We have glycans, and the glycans and the motifs obviously are connected. There are 33,000 glycans. They are connected to the proteins either by binding or by the site, which means they are glycosylated. So there are 64,000 proteins. We also have disease, organisms, and genes, and that's all presented in that graph. Now, what you can do is you can click on any of these boxes and you can perform a query on that. So for motif, let's say we are looking for a motif with the name Lewis X. And now, my bad, Lewis X. It will search for the motif with Lewis X, but then it will update all these boxes which are connected to that motif and only show information which is related to that search query. So now we can see there's only one motif left with that name. From the 33,000 glycans that we have, 1,600 actually have been reported to carry that motif. And if I would click on that little yellow box here, it would actually get me to the list of glycans which have that motif. But what you also can see is that there are only 86 proteins that have been reported associated with that motif, because we either bind glycans carrying that motif, or we have glycosylation sites where one of these 1,600 glycans have been reported. So that really allows you to do a query from one of the concepts like motif and even find proteins which are not directly associated with that motif, but have that indirect connection via the glycans. So these are the searches that we implemented. There is one more search I would like to show, and that's the so-called site search. What you can do here is you can look for proteins based on their sites, glycosylation sites, but also mutation sites. So let's say we look for a site which happens to be on the asparagine. I could click on search now and it would show me all sites which where an asparagine is involved. But I can also refine that search by saying, okay, find me all asparagines which have been reported to be glycosylated and which have single nucleotide variations and mutations at the same site. And if I'm clicking on search now, it will go through all the several 10,000 sites that we have for all the proteins in our database, and then only show the sites for the proteins that have an asparagine which is reported to be glycosylated and has a mutation reported as well. And then by clicking on the protein ID on that list page, you actually can go to the protein and look into detail what's going on there. Now, what I would like to do next is that gives you a good overview on what kind of information we have in the database and how you can search and find the glycans or proteins that you are interested in. And I encourage you to go to the web page and do that. But in the rest of the talk, I would like to introduce a bunch of new features that we have developed in the last few months and which we will release by the end of the month to the public. You can already access them on the beta website though. So the first one is I'm going back to the glycan page. And on some of the glycan pages, you have that little button here, which says Sandbox, which is one of the satellite projects that we have. And by clicking on that button, you actually go to the web page of that project, and it shows you information about the same glycan that we just looked on GlyGen, but it shows you slightly different information. So we have here the list of residues that are part of that glycan. We also can see a list of enzymes that may have been involved in that glycan, but just pretty much the same information that we already saw on the GlyGen web page. And we can see the related glycans, which are close to that structure. But what I want to focus on is on the pathways and the enzymes. So if you click on the Show Pathway button, we will get a different view. And what we see here is that each row corresponds to one glycan. So that's the N-glycan complex core in the first row. And then we have multiple different structures. And in the end, we have the structures that we actually have looked at. And all the structures in between are potential candidates for being part of the pathway that may have led from the N-glycan core to that complex structure. And you can also see these arcs on the right side here, where each of the arcs represents a single reaction that adds one residue to the structure. So that's essentially your pathway, step by step, building that structure. What users can do is we can use that little tick box here to essentially build the pathway that we're interested in. Let's say we want to be that structure be part of the pathway. We click that tick box, and then we have a pathway from the start to the end. And then we can render this pathway and see all the structures step by step. How do you build that complex structure in the end? For each of these steps, you can see the monosaccharide that has been added to the structure. And you can also see the list of enzymes that may have been involved in catalyzing that reaction, adding the monosaccharide. You can also filter by the species and just show the human enzymes, or just show the mouse enzymes for that corresponding pathway. So that's one of the tools that you can access using that sandbox button from the GlyGen details page. Another thing I would like to show is the search results for glycans and proteins. Just we're going to add a new feature that if you perform a search, for example, in this case, I searched for all human proteins, and it has a list of 20,000 proteins. But then on the left side here, you can see filter options that you can use to refine your search further. So let's say we go to the end of the list, we have that 20,000 human proteins, but we only want to see the proteins that have been reported to be phosphorylated. So I'm clicking in the PTM section on phosphorylation, and then out of the 20,000, only 10,000 are left. And then let's say we also want to have the proteins that at the same time have been reported to not just be phosphorylated, but also have a O-glycosylation. I click on the O-glycosylation with the phosphorylation also selected, and now we have 135 proteins left in our database, which have phosphorylation and O-glycosylation sites. You can also further refine it with other of these groups. For example, by saying, okay, I only want to see the proteins that have a disease relationship, or only see the proteins that have glycan binding reported. The same kind of list is also available, or a similar search list is also available for the glycans, where we perform a glycan search. We get the list of glycans, and then on the left side we have other possibilities to filter that. For example, we can only look at the glycans which have papers attached to them by clicking on that publication, or we can look at glycans which only have specific monosaccharides or substitutions, or we can also just click on fully defined, and then it will only show us the list of the fully defined structures which have no missing information. The last feature I want to show you is the alignment. So on a protein details page, we can go to the list of homologs, or also the list of isoforms, and then we can align them with each other, which gives us a sequence display where we see the different sequences from human, mouse, and rat in an alignment or standard alignment display. But then on the right side here, we have that sites button, which allows you to show specific sites on these proteins. So for example, let's say we want to see all the N-glycosylation sites and the N-glycan sequence. So down here, we can say, okay, we have N-glycan sequence here, 4 of them, and we can also see that these 4 are actually also reported to be glycosylated in all 3 species. But then if we add the O-glycosylation site, we actually only have a single site right here. And what we can see is that only in the human, this site has been reported to be glycosylated, but not in mouse and in rat. So that's a very convenient way to compare the different homologs with each other. We also have an option where the user can enter a custom peptide and highlight that custom peptide, and also see if the same peptide is still present in the different homologs in the different species. The last thing I would like to mention is if you're going to the web page and using the web page, you're playing around with it, there's something that you noticed which is causing problems, or if you have suggestions, please use that feedback button, which is pretty much present on all web pages, and just send us a message with the changes you would like to suggest or the problems that you found, and we will be working on them. With this, I would like to finish my talk. Thank you for your attention, and please feel free to go to our webpage and use it for your research. Thank you.